Okay, so now let us start for today's lessons. I'll talk about these generations of action potential. Now, we do know that for neurons, the function is to transmit the electrical signal because the nervous system uses the electrical signaling pathway, okay, as contrast to the chemical signaling pathway which we use the hormones. So this signal travel or conduct very rapidly along a cell surface membrane. Actually, it's not in the cytoplasm, but at the cell surface membrane from one end of the cell to another end within few milliseconds. Okay, but the signal are not flow of the electron like the electric current. So it's a little bit different from the electric current. Electric current is the electrons, the one that keep on moving, right? But for this action potential, it's not. Okay, it's not. So we're going to learn in the next lessons how this action potential can be transmitted. Okay, so the signal are very brief changes in the distribution of electrical charge across the cell surface membrane, known as action potential. And this because of the rapid movement of the sodium ions and potassium ion into and out of the axons or the neurons. Okay, so before we start, okay, to look at how we can generate the action potential, then first thing we need to learn the resting potential. Okay, the resting potential. So what does mean resting potential? The resting potential is a potential difference exists across every neuron's plasma membrane. And we use this, the electrode to check, and you will realize that there is a membrane potential of negative 65 millivolts. If you use this electrode to check, one electrode you place it on the surface, another one you place it inside the cytoplasm, then you can see that there is a membrane potential of 65, the membrane difference or potential difference of 65 volts or millivolts in this case. And the negative actually refer to the internal, okay, surface of the membrane. So this internal surface of membrane will be negatively charged and outside will be positively charged. So it gives us a negative 65. And this potential difference is known as the membrane potential. At negative 65, we give them a special name called resting potential. Okay, it's known as resting potential. Here. So how we can maintain this resting potential? So what's so important about this resting potential? Because any neurons, before we can stimulate the neuron must be maintained as negative 65 millivolts. Always maintain at this negative 65 millivolts. Okay. So the inside of the cells is more negatively charged in relation to outside because of three factors. Three factors, but the most important factor highlights number 2 and number 3, which involve the sodium potassium pump, actively pump 2 potassium ion into the cells for every 3 sodium ion pump out. And also the ion channel allow more K plus ion to diffuse out compared to the sodium ions. So therefore, with these two mechanisms, we will be able to establish the membrane potential roughly about negative 65 millivolts. Later, I will show you, okay, how we can achieve this membrane potential. So let us go through the notes first, then later we can see, okay? So sodium potassium pump actively pump out 3 sodium ion for every 2 potassium ion move into the cells, and the process involves ATP. So means that you need to use up a lot of ATP to carry out this process. So this process involves moving the ions against their concentration gradients. So therefore, it maintains a resting potential by keeping more sodium ion outside the cells because we pump out 3 sodium ion, while more potassium ion inside the cell because we continuously take in the potassium ions. Okay. And at the same time, okay, so the neuron membrane is more permeable to the potassium ion compared to the sodium ion due to the presence of more potassium ion non-gated channel than the sodium ion's non-gated voltage independent channel. So in our syllabus, you just term it as the ion channel enough already. At this for this one, you just name it as ion channel. Okay, so therefore, because we have more potassium ion channel, definitely more potassium ions can diffuse out of the cells by faster diffusion compared to the sodium ion that diffuse it. So overall, we're going to have excess positive ion outside the membrane compared to inside. Then we say that the membrane of the neuron is now polarized. Are you clear? So membrane is polarized. Okay, so let us look at the process. Okay, so now look at this. This is a neuron. This is the terminal at the end of the neuron. Okay, so now we're going to look at these different ions involved. So we have few ions. So we have the concentrations of the anion, negatively charged ion. We have the concentrations of the potassium ion. We have the concentration of the sodium ion. This one is outside of the cells. Okay, outside of the cells. And inside of the cells, we do have another set of the anions, negatively charged ion, potassium ions, and sodium ion. Okay, so I give a figure, okay, this make up the figure, okay, so their concentration per unit, okay, how many sodium ions, potassium ions, as well as the anion outside the cell. So imagine that now neutral. Neutral means that the number of the positively charged ion equal to the number of negatively charged ion. So let's say outside, let's say here 500 units. Potassium ions, let's say 300 units. And ions, because neutral, right? So total up 800 units of anion. So balance the charge. So now 0. No positively charged extra, no negatively charged extra. So inside the neuron also, we have another inside neurons. So let's say 200 units of the sodium ions. Okay, potassium ion, let's say 400 units. Are you clear? Okay, 400 units. So total up here 600 units. Correct. So means the anion 600 units. So at this moment, can you see that they are neutral at this moment? Correct not? Okay, the number of positively charged ion and negatively charged ion, they are the same. Okay, so now in order to get to the resting potential, we depend on two mechanisms. So we look at the first mechanism, which is a sodium potassium pump. So what we need here for sodium potassium pump, we actively pump out 3 sodium ion. So by using the ATP to phosphorylate this sodium potassium pump, then we can see that actively we pump out 3 sodium ion. Take in 2 potassium ion. Now, this is one round. What will happen here? If we do a simple calculation now, okay? Simple calculation. Do you realize that now it's no longer neutral? Now, why is it no longer neutral? Look at this. First of all, okay, look at this. First of all, sodium ion now, because we pump out 3, so outside now no longer 500 unit. It add more, so 503. Inside, because we pump out sodium ion, again the concentration gradient, right? Inside 200, outside 500, we pump out. So again the concentration gradient, so no longer 200, now become 197. Correct? You get the idea? Okay, because we pump out 3 sodium ion and take in 2 potassium. So when you take in 2 potassium, outside now no longer 300, it's 298. Correct? Potassium ion inside now become 402. So if you look at the positively charged ion inside would be 801. Correct not? Okay. And the positive ion inside the neurons here, you can see that is 599. So if you look at this, the calculation now, so outside now start to have a positive 1 charge, inside now -1. So you start to build up. Can you see that? It start to build up this already. Positively charged outside, negatively charged inside. So for every round, okay, can you see that? For every round of the sodium-potassium pump, so we constantly pump out 3 sodium ion, pump in 2 potassium ion. At the end, you can see that outside now become more and more positive, inside become more and more negative. So this is the first mechanism. Okay, how about second mechanism? Second mechanism, the neuron actually is more permeable to the potassium ions compared to the sodium ions. So let us look at this. 1, 2, 3, 4, 5. Okay, 6. So you can see that they have more potassium ion channel compared to the sodium ion channel. So green color, sodium ion channel. Okay, blue color, potassium ion channel. So means that you can see that, guys, inside potassium ions 402, outside potassium ion 298. And this is ion channel. So potassium ion now leak out, and leak out with a higher rate compared to the sodium ion. Can you see that? Potassium ion now move out, compared to the number of the sodium ion that move in by facilitated diffusion. So now we continue to calculate. I'll remove this one first. Now, if we continue to calculate, right? So outside, can you see that now we have extra 1, 2, 3, 4, 5 potassium ion. So 298 now plus 5, 303. Okay, can you see that potassium ion? So inside 402, we leak out 5, right? We leak out 5, so -5. So 397. Okay, 397, leak out 5. Sodium ion from 503 now become 502. Can you see that? And then sodium ion inside, we add another one. So 198. So if you calculate now, you can see that outside, positive ion become 805. So overall, positively charged 5. Can you see that? Have more positively charged ion outside. And here you can see that 595. So overall inside become negatively charged. Can you see that? So if this thing continue on and on and on, then you can see that now membranes, because of these two mechanisms, what will happen here is the membranes will have the membrane potential. This membrane potential will be established as -65 millivolts. So try to imagine if this process continue and continue and continue. So it means that it will start from the 0 millivolts and go all the way down to -65 millivolts by having these two, okay, mechanism. But can it be more than -65 or not? The answer is yes, but slowly, because outside is too high in terms of the positive number of positive ion, and inside too high in negative ion, then you can see that electrochemical gradients actually make sure that we maintain the resting potential at -65 millivolts. At -65 millivolts, we say that the membrane is polarized. The neuron membrane is polarized. Are you clear? Because one side positive, one side negative. Outside positive, inside negative. Okay, so this is how the two mechanisms, okay, two mechanisms that help us to establish, okay, the membrane potential. Okay, membrane resting potential at -65 millivolts. Okay, so I repeat again a summary here. So how our neuron maintain the resting potential? They maintain the resting potential by using these two mechanisms. So the first mechanism, how they do it? They use the sodium potassium pump. So ATP actively phosphorylates the sodium potassium pump. So sodium potassium pump constantly pump out 3 sodium ion and take in 2 potassium ion. So by doing this, you can see that they have extra positive ion outside the cells, outside the neuron cells, compared to the inside of the neuron. That's first mechanism. Second mechanism, you can see that the neuron is more permeable to the potassium ion compared to the sodium ion. So it means that the potassium ions will move out of the cells in a higher rate. Because why? Because of the sodium potassium pump. Sodium potassium pump always makes sure that potassium ions accumulated inside the neuron. So it means that with a high concentration of potassium ion inside the cells compared to the outside the cells, therefore more potassium ions will move out of the neurons per unit times compared to the sodium ions that move in by facilitated diffusions. Okay, so I want to write it down here so that this symbol basically is the potassium ion channel. Okay, and green color, this is the sodium ion channel. So non-gated, okay, always remember non-gated voltage independence. Okay, so means that facilitated diffusion take place. So this part involves active transport. This part involves this facilitated diffusion. Okay, so you can take a photo if you need, or take a screenshot. Then you don't need to draw it out. Okay, done. Okay. So stop the recording.